Good day to everyone joining us, and welcome to today's Xtalks webinar. Today's talk is entitled Streamlining Cell Line Development for Faster Biologics Production. My name is Sonia Hunt, and it's my pleasure to be your Xtalks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and comments for our speakers throughout this presentation by using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, which is found on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. I'd also like to welcome our attendees viewing this presentation from our LinkedIn live event, and I encourage you to submit questions as well via the comments tab. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming to those who register on xtalks.com. At this point, I'd like to thank ATUM, who developed the content for this presentation. ATUM has a 20-plus year history of innovation. ATUM provides services for biologics, drug discovery, and development, including gene sequence design and synthesis, antibody engineering, and transient production through cell line development. They use advanced AI, machine learning, and proprietary algorithms to de-risk and accelerate time to market for biologics. Now, I'd like to welcome our speakers to today's event. And first, I'd like to introduce you to Claudia. She is a trained pharmacist and obtained her MSc from the University of Coimbra. There she is, in Portugal. She started her career at a CDMO with a focus on cell line and cell culture development. Claudia joined the USP team at CDR-Life over 2 years ago and is now the lead scientist in the drug substance group. She specializes in developing a USP platform for CDR-Life's proprietary mGager T-cell mGager format. Now, I'd like to welcome Lynn. She is a seasoned expert in mammalian cell line development, and there she is. Hello there, Lynn. Over the last 8 years at ATUM, she has established herself as the go-to expert on all practical aspects of Leap-In technology applications and is a co-author on seminal papers on the use of the platform. Now, with the introductions out of the way, it is now my pleasure to pass the mic over to Lynn. So, Lynn, when you're ready, you may begin.
Hello and welcome. Before our guest speaker comes on, I wanted to give you a little background about ATUM and our Leap-In technology for cell line development. First of all, ATUM has been in the business for over 20 years, initially called DNA 2.0. When we decided to expand to include cell line development services in 2016, we changed the name to ATUM, which means the Egyptian god of everything, to encompass all of what we do. On the left is our headquarters, where we have services extending from gene synthesis to protein expression and protein engineering. And on the right is our building 8484, which is just down the street, that opened in 2022 and is fully dedicated to cell line development. Both of these buildings are in Newark, California, and are 100% solar powered, as you can see by all the panels. And all of our work is performed here. What spurred our decision to start providing cell line development services was the invention of our Leap-In technology. Leap-In has been used to produce stable cell lines for at least 45 IND filings. Our clients have filed in 4 jurisdictions so far: the US, the EU, China, and Australia. We have over 60 licensees for the technology, and we have delivered over 400 cell line development projects. These include cell line developments all the way to cloning and pool projects. The breadth of our projects reflect the complexity of molecules and therapeutics. We've run projects on all sorts of molecules, from antibodies to bispecifics to vaccines. While we're not a CDMO, our expertise in drug designs helps address molecule limitations while we're developing the cell line. We also support technology transfer to help our clients troubleshoot during scale up and navigate regulatory issues related to the cell line. our work is enabled by Leap-In. So let's discuss why so many projects have come to us. First and foremost, the benefits of Leap-In technology are that we can manufacture robust cell lines with stable clones and high titers. Because those titers, because of those higher titers, more can be done at an early stage, at the early bulk pool stage. And Claudia will go through some examples of that in her talk after mine. Just briefly though, transposase is an enzyme that we've engineered to optimize integration, but our gene editing capabilities start with the vector. We've also optimized transposons, aka the DNA constructs, to accommodate multiple open reading frames that simplify the cellular production of complex biologics. So how does Leap-In work? We start with the transposon vector that's up on the left-hand side there. The expression cassette is flanked by specific inverted terminal repeats, or ITRs, that are meant to interact with the TTAA motifs conserved throughout the host genome, which is on the right-hand side there, in potential transcriptional hotspots. So we co-transfect the cells with Leap-In transposase mRNA, which recognizes the ITR and excises it, and then integrates the full expression cassette into the target sites in the genome without creating concatenamers or partial integration. So, as for the transposon itself, multiple open reading frames in a single expression cassette can be predictably expressed in designed ratios for maximum intact molecule production. This is one example shown here of a bispecific with 2 open reading frames, one in pink and one in blue. By engineering the transposon, we can control the relative expression of one chain versus the other. Complex biologics such as bispecifics don't necessarily express to an even degree, but Leap-In gives us the flexibility of testing various ratios of the open reading frames, and it allows us to optimize expression in the bulk pools that can be carried forward into clones for manufacturing. Okay, so Leap-In has also given us another benefit. It has dramatically reduced the number of clones to screen for high producers. On the left-hand side, we have a cartoon of random integration where it would take screening a stack of 10 ELISA plates, nearly 1,000 clones, to get a very few number of high producers, which are represented here by the blue bar. And then the productivity of the clones tapers off drastically as represented by the pink line. On the right-hand side, we actually have data from the Leap-In cloning campaign where you can see the original pool in the middle in orange and the entire clonal distribution, half are high, half are lower than that clone in the middle. So we have a large number of very good clones to choose from. And this is a screen of only 100 clones. So the Leap-In pool is giving 1,400 megs per liter in this example on day 7, and the top clone is giving 2,400 megs per liter. Imagine what they could produce in a 14-day fed batch. So we don't have to imagine. Here are some of the ranges of productivity that you can see that we've recorded for some of our more common molecules that we've expressed. Please note that the lower end of the range are unoptimized shake flasks used for pool and clone screening purposes only. The higher end numbers we've gleaned from optimized processes either here at ATUM or our cell lines in the hands of a CDMO or CRO after process optimization. So you can see that, some of them are quite high. So cell line development for bispecific antibodies. So we had put together a paper that came out in August called Cell Line Development for Bispecific Antibodies, Better Predictability Through Transposases. So this slide on the left-hand side shows volumetric productivity, and the right-hand side is percent heterodimer. And so what we're looking at here on the left side is a consistent 4 gram per liter from pool to clone, from population doubling 0 to population doubling 60. So PD0 is in the blue, PD60 is in the pink and the yellow, plus and minus glutamine, which is our selective agent here. And the same is true for the heterodimer. It's consistent in the range of 75 to 85% from the pool to clone, and also from PD0 to PD60. So the pool predicts the titer, and the pool predicts the percent heterodimer and the molecular characterization of the clone. So this is a schematic of our cell line development process. It's a transfection to RCD in 13 to 19 weeks. So what we want to emphasize here is the power of the pool. So if you see that green line in the middle of the range, there is where we get the pools. So the pools give us an opportunity to quickly evaluate different constructs, ratios, et cetera, whatever we want to look at. And characterizing our pools gives us an early read on product quality via analytical development. It can allow time for upstream and downstream process development, and allow for tox lot generation, all of which de-risk later clone characterization, moving the project ahead on a critical timeline for manufacture. So this is the end of my slides. Thanks for your attention. I'm going to hand off to Claudia for her presentation involving CDR-Life's experience with our Leap-In pools.
Thank you, Lynn. I hope you are all seeing my slides now. So first, I'm going to introduce you to CDR-Life. CDR-Life is a clinical stage biotech company at Zurich Lake. It was formed in 2017, initially in Schlieren. However, the company has grown, and we moved to a bigger facility in Horgen in the Zurich Lake. We have developed a successful partnership on CDR-Life assets with Boehringer Ingelheim. Our technology is based on mGauger platform, and it's basically a molecule that is modular, stable, soluble, and manufacturable that can target basically any antigen. In our pipeline, we have several assets, as in CDR-404, that is candidate that is right now in clinical trials phase 1, and CDR-609 that we are currently developing that targets colorectal cancer. So what is an mGauger? An mGauger is basically a T-cell engager that targets clean antigens and is a bi or tri-specific T-cell engager. So basically, our mGauger can target 1 or 2 epitopes of the cancer cell while engaging the T-cell for killing the cancer cell. In CDR-Life, we have means to go from end-to-end platform, so basically from idea to clinic, and all of this has allowed us to build a diversified pipeline. So we have several assets in our pipeline. 2 of them are already in clinical stage. As I mentioned before, we have end-to-end platform here at CDR-Life. Part of that is also due to our deep CMC know-how in our CMC team, where I belong. We have specialized sub-teams that cover all from cell line development to clinical manufacturing. So cell line development is the topic of this talk here today. We do it with ATUM. And we basically have synchronized cell line development approach here. So basically ATUM creates our cell lines for us, but the stable pool evaluation is done at CDR-Life, and also this is also able to coordinate activities between the CDMO and CDR-Life. Our upstream team is also responsible for producing material for PK and PD and tox studies, and also to provide material for development of other departments, and also to do process development for transfer up to 50-liter scale in-house. Our CMC team also has other teams such as downstream processing, analytics, drug product, and clinical manufacturing. So let's focus on upstream. And I'm going to show you how our platform works from discovery to IND. We have usually, essentially we split our upstream development within 3 phases. So stable CHO pool, monoclonal RCB, and master cell bank. Right now, we focus on the stable CHO pool. So on the stable CHO pool, our main goals are to establish a platform fit of the recombinant CHO cell line, do the CHO pool evaluation, lock already the culture medium and feed. This is super important because we need to define early on our culture medium and feed and communicate it to our CDMO, so we are sure that it's going to be available for our manufacturing campaigns. We also lock culture parameters and also define the critical quality attributes. All of these steps allow us to get representative material for tox and preclinical studies already from the stable CHO pool. So none of this would be possible without having developed a platform. Otherwise, it would take us too long to test all of the different conditions. And developing a platform, it's only doable if we have constant cell line. So for that, we have established a collaboration with ATUM. We have over 6 joint engager projects, and ATUM contributes with a robust CHO-K1 cell line with the Leap-In Transposase system. So we have our cell line, and also luckily our format is rather similar between projects. So basically we did a huge screening with different media and different feeds over multiple cell lines, and we ranked the results with by titer and protein quality. And after all of this work, we have achieved our top 3 conditions that are basically preferred condition or a go-to condition that would yield us very high titer, and 2 backup safety conditions that are basically conditions where we compromise a little bit more on the protein titer, but we do get higher protein quality, when actually protein quality is one of the issues that you want to tackle most. So after we developed, so early on we developed our platform process, and this is basically how our upstream development using stable cell pool looks like. It starts with the transfection at ATUM. So ATUM transfects the cells with our construct or constructs and selects the cells, and after the selection is done, ATUM ships the stable cell pool to us. This for us is super important because we need to plug in our platform. So usually we do a starting run with our 3 top conditions that I mentioned before on the slide before for the platform, together with ATUM condition as a control. And we evaluate here the different molecules that we want to test and different also constructs that you want to test for each molecule. And after the evaluation, we select our preferred condition, so our media and feed selection that we later on communicate for our CDMO. Here, as you can see, we decided to go with condition number 1. That is our generally go-to condition that has given us higher titer and okay protein quality. After we select our temperature shift, so for this we do a study, we use DOE because here we really want to get the optimal window for doing the temperature shift and the temperature after the shift, obviously taking into account our main target here. In this case, we have defined the critical quality attributes as being aggregate level. So we lock then the top conditions, and this obviously led us to our final goal, that is to get representative material from CHO pool, and we use this material in our PK/PD studies and tox studies, and we know it's going to be very similar to our final product. Then we get to our next stage, it is the monoclonal RCB. Here we do a clone selection joint with ATUM. We also do process and feed optimization to top clones. We do upstream process lock and scale up, and tech transfer to CDMO. The main goal of our monoclonal RCB is to have a completely optimized process before process lock. So we don't have to worry more about it. We have a smooth tech transfer, and we know that we are going to achieve IND submission with minimal risk. So how does our clonal selection look like, especially if it's done between the US and Switzerland? This all starts by ATUM. So ATUM clones the CHO pools and picks 48 clones, and there's the expansion of these 48 clones, and later on a first fed batch evaluation, a first 14-day fed batch evaluation. After the harvest, ATUM ships the supernatants to us, where we do a very thorough protein quality analysis. So here we take leverage of all of the biological data that was provided by ATUM, such as growth, viability, also protein titer, and we also take advantage of our data from the purification of these supernatants. And then all of our CMC team gathers, and we look at the results. Here, as you can see, aggregates was our critical quality attribute. So here we have actually plotted our top clones and how does it look like between aggregate level and purified titer. Then later on, we select our top 24 and 12 clones. ATUM proceeds to the RCB generation of the top 12 clones and a confirmation run of the top 24. The confirmation run of the top 24, we have the supernatant also shipped to us. Again, one of the things that we are looking for is robustness. So this also allows us to select our top 5 clones with some safety, that we know that most likely we are making the right decision. So our next step, we select the top 5, we get the RCBs from ATUM, and our first step is to transfer to bioreactor. So here we plug in our platform process, everything that we have defined before in the stable CHO pool phase. And we see if we need any kind of optimization or improvement. As you can see here on the lactates graph, we did need improvement for this project in particular, because we had a very big difference in the metabolic profile comparing control shape flask and the bioreactor. And then we select our top 3 clones, and based on the results that we had on the first run, we run a feed optimization, trying to improve the process, leading us to a final clone and the final locked process. As you can see here, this is the lactate profile of the exact same clone as I showed you before after our feed optimization round. Here we did 2 bioreactors in parallel for our process lock, together with the control shake flask, and as you can see, the results are very similar. So this led us to our last phase, that is the master cell bank. On the master cell bank, we have the biosafety testing, mock master cell bank generation, master cell bank creation, and conditional release of the master cell bank for manufacturing. So basically, all of this will allow us to reach IND smoothly with minimal risk. So we don't do the master cell bank in-house, as we don't do the cell line development in-house. So how do we make it work actually? So for this, we have to create parallel work streams from cell line to IND basically, and there's a global collaboration between ATUM, CDR-Life, and our CDMO. Our CDMO can be anywhere basically. So all of this starts with a research cell bank created by ATUM. So ATUM ships us the research cell bank of the top 12 clones. Then while we test all of the clones through the phases that I've told you before, like clone selection and process development, ATUM also coordinates the shipment of the RCBs to our CDMO. Then again, here we are doing clone selection, process development, and process lock. By the time that we do the process lock, we are already sure of our top 3 clones at least. So we start biosafety testing and all of these clones in our CDMO to minimize delays, because biosafety testing takes quite some time. So in order to take advantage of this whole time that we have in-house, that is our process development, is absolutely necessary time. We do the biosafety testing of more than what we need. So after our process is locked and we have selected our top clone, we go for master cell bank creation and characterization. And as we both know, this is a very long period, and at the same time, we start the process of transferring the process to our CDMO and scale up at the CDMO. In order for when we have our conditional release of our master cell bank, we are ready for clinical manufacturing, and therefore IND submission without any delay and very smoothly. So to show you that actually these are not just ideas and it actually works, we have established our successful clinical manufacturing campaign for CDR-404. We achieved a robust and scalable process from 3 liters to 500 liters for GMP manufacturing. As you can see here, SEC HPLC purity is very similar in between the different scales. Also, VCD profile, very similar in between the scales. So it was a very successful scale up. We have successfully submitted IND in December 2023. And currently we are in clinical trial phase 1. Any of this would be possible without the help from everyone. And I want to give a special thank you for the CMC team of CDR-Life. It's really necessary to cut timelines that we have a team working together to achieve the same goal. So thank you very much to all of the team. And that's it. Now over to you, Sonia, for Q&A.
Well, thank you very much, Claudia and Lynn, for that great presentation. I hope everyone enjoyed it. And now we're going to begin the Q&A session of this webinar. Now, if you still have the opportunity to send your questions and using that chat box, so please go ahead and do so. And if you are attending this via LinkedIn Live events, if you're watching it there, please utilize the comments tab. Okay, and we'll try to get to your questions during the time we have together. So let's start with this first question here. This question looks like it's for you, Lynn. For customer projects, do you determine vector design in advance or test many vectors simultaneously? A great question. What are your thoughts there, Lynn?
So we have a team of project managers who also do the gene design. So they have a lot of interaction with the client up front to talk about what the client wants and what their needs are. And they do quite a bit of gene design. They'll come up with multiple strategies. So we do quite a number of pools depending on what the client wants, just so we can narrow down the field of what works and what doesn't for the client.
Okay, thank you, Lynn. Let's go to the next question here. Oh, this is for you again, I believe. How many copies of the gene of interest are generally inserted into the genome?
Okay, it really depends on the construct system that we're working with. It can be up to 60, but sometimes we'll have very good results with something that makes, has 8 copy numbers or 4 copy numbers. There isn't a real, one-to-one correlation between the number of copy numbers and the amount that it actually makes.
Okay, thank you. I'm here busy getting all these questions that were coming in from our audience members, so just give me 1 second here as I try to attend to all of them here. Okay, all right, so let's go to the next question here. This looks like it's for you, Claudia. What are typical variables like pH or media type that you look for in the process selection?
So first, for the platform development, actually, obviously we look into chemically defined media, good for manufacturing, and that we can actually, that has a very good distribution worldwide. But for really process development, I look into more like the growth of the cells, the metabolic profile of the cells. This is super important, and and protein quality essentially.
Thank you very much. Okay, let's go to the next question here. How many copies were integrated to achieve 4 grams per liter of the MAB, Matt?
Right. So
Lynn is smiling.
It's probably in the paper, so I don't want to say like quote an actual number because it's probably out there. But I would imagine it's you know, in the tens, probably between 6 and 12 or something like that.
Okay, thank you very much for that. Let's go to the next question here. Let's see here. Okay, can you put a timeline to the RCB to IND scheme presented in slide 21?
So this is a work done basically, I mean, we in the company, we have had several projects. Within each project, we do learn a lot of things and where to cut corners, where to de-risk. Right now, we are at the point that we can reach IND from monoclonalization in around 10 months to a year, if I'm not mistaken.
Okay, thank you for that. Give me 1 second here. There's so many questions coming in. I'm trying to attend to them. So give me one moment as I get everything all organized here. All right, so let's go to the next question. Let's see here. This audience member is asking, oh, for Claudia, are you exploring post-translational modification like glycosylation as part of the process development?
We are very lucky that our molecule does not have FC fractions, so it doesn't have any glycosylation. Typical one, we do observe some O-glycosylation in the molecule, but we have seen also that it's safe and the molecule is still active. So we don't really have to worry about it.
Okay, thank you for that. Okay, let's go to our next question here. What are typical variables like pH or media type that you look for in the process selection? I think that's again for you, Claudia.
Yeah, I think this one I have already
Oh, you did?
answered.
Okay, they put it in twice. All right, no problem. Okay, let's go to this one here. How many copies of the gene of interest are inserted into the genome? Did we do that one yet?
I think already, yeah.
Yeah, okay, so it looks like we've got multiple questions coming in from various people. Okay, let's go to this one here. I think that's all the questions for now. I think we got everything here. Oh, there's one more, sorry. How do process time differ between the transposase mediated cell line versus a non-transposase mediated cell line?
So random integration versus transposase. It really, because of the screening, the intensive screening we would have to do for the random integration, that takes a lot more time to go through all that, to get all those clones. So yeah, so Leap-In also gives us this pool that we can use to do all this upstream and downstream scouting. So that compresses our timeline as well.
Okay, thank you so much for that, Lynn. Let's jump to the next question here. How much time could be saved by CDR-Life by designing or assessing the USP in-house versus having it done directly at the CDMO? And what are the main advantages you see? Claudia, that question is definitely for you.
Time saved. I mean, obviously, as you know, out there are companies that promise everything within even like 6 months. But there's obviously an advantage of doing everything in-house. First of all, we do know our molecule better than any CDMO. So we know what we should look to. We also have a format that is very similar in between projects. So we do save a lot of time because we already can predict what's going to happen and we already know how to tackle all of these issues. So I would say that it's all of the knowledge that we do have in-house regarding our molecule. And that's obviously an advantage than just transferring everything to the CDMO.
Thank you for that. Let's go to the next question here. And this question here is, how does pool productivity and PQAs compare to clones when using Leap-In Transposase versus this targeted integrated platform?
We would have to test it against the targeted integration platform, which I don't think we have that data.
Okay, thank you. Let's see what the next question is. Sorry about that. I'm just looking here to see what we have here. Okay, I think that's it so far. Let me just see if we have anything else coming in here and anything is approved. Okay, all right, I think that's it. We have a lot of other questions that have come in from our audience, but those are going to need a little more detail, and we don't have enough time to go through all of them. So I just want to say, if your question was not answered, the team will follow up with you after this presentation. So that ends the Q&A portion of this webinar. Now, if you have any further questions, please direct them to the email address you see there on your screen. Thank you, everyone, for participating in today's webinar. You will be receiving a follow-up email from Xtalks with access to the recorded archive of this event, and a survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. Additionally, give me a few seconds. I'm going to share a link to view the recording of today's event in the chat box, which you can share with your colleagues once they register for the recording here as well. And for those of you watching via LinkedIn Live events, we encourage you to visit xtalks.com to register for this event and gain access to features such as viewing the recording and more. Now, please join us in thanking our speakers for their time here today. So thank you very much, Claudia and Lynn. It was very inspiring and very thought-provoking. So thank you very much for your time. We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at Xtalks, we thank you for joining us. I'm Sonia Hunt. Until we meet again, please take care and bye for now. Bye, everyone. Bye, Lynn. Bye, Claudia. Bye. Bye.